name is Natalia from São Paulo State University, and I will present the work Fluorescence Activity and Structural Response of the Enhanced Green Fluorescent Protein, EGFP, to pH, Potentials for Biosensors. This work was published in the International Journal of Biological Macromolecules in 2020. EGFP is a remarkable protein that has intense and quantifiable fluorescence and is compatible with biological systems. Because of these properties, it can be applied as a biosensor and biomarker. Recently, there is an increasing effort to develop biocompatible pH biosensors for the medical area, to monitor conditions with pH alterations such as tumors and synovial diseases. However, EGFP application in this field is still limited by the lack of information on its behavior as stress conditions. For example, there are already studies on the range of EGFP fluorescence activity at different pH. However, further studies are necessary to demonstrate the reversibility of EGFP fluorescence quenching and to do a structural analysis of the full process. Hence, the goal of this work was to evaluate the effect of pH on the fluorescence activity and structure of EGFP to assist in the development of pH biosensors. In this work, we exposed EGFP to different pH values for 30 minutes and evaluated its activity by fluorescence, its microenvironment changes by intrinsic fluorescence of proteins, its secondary structure by circular dichroism, and its tertiary structure by small angle X-ray scattering with and without size exclusion chromatography. After this first evaluation, we returned the pH to neutral for 60 minutes and did another assay. At first result, we can see that EGFP was stable from pH 7 to 13. From pH 6 to 5, there was a partial loss of fluorescence, and from pH 4 to 2, there was a total loss of fluorescence. Later, we studied the reversible effect of pH on EGFP, and we can see that at pH 6, there was a total fluorescence recovery of the fluorescence with the return to neutrality, and only a partial recovery from pH 5 to 2. In the circular dichroism analysis, from pH 6 and above, we did not detect any alteration in EGFP secondary structure, but from below pH 6, there were structural alterations, especially a decrease in beta strains and an increase in unordered forms. And this effect was only partially reversible with the return to pH 7.4. In the Saki-Sachs study, we can see that from the pH 7.4 to pH 6.5, there are also no alterations in the tertiary structure of EGFP, and the differences in fluorescence was caused by only a protonation of EGFP without irreversible structural changes. However, from pH 6.5 to pH 5.5, there were irreversible structural changes in the protein, particularly some loose strands. In the last study, we also evaluated EGFP fluorescence intensity versus pH, and we could accomplish a sigmoid curve with R squared above 0.99, and we could see that small variations of pH produce discernible variations in fluorescence intensity. These two properties are allied with the reversibility of EGFP quenching, are very good properties for pH biosensors. Hence, we can conclude EGFP is highly pH dependent, with reversible loss of fluorescence from pH 6 to 7, fluorescence quenching and structural modifications below pH 6, and small variations of pH produce discernible variations in fluorescence intensity of EGFP. Additionally, the Saki-Sachs was useful to determine the structure of GFP in a complex environment, and that finally, EGFP has potential application as neutral to acidic pH biocompatible biosensors. Thank you very much for your attention.